Hello everyone. Today we are going to discuss chemical synapse, okay, and how the nerve impulse conduct through the chemical synapse, okay, and what is the role of neurotransmitter in the chemical synapse or nerve impulse conduction. So, chemical synapse, as you to see in this figure, there is three different types of synapse may be there, okay, and as you to see in the figure, there is a presynaptic neuron, okay, which receive information, and a postsynaptic neuron, okay, which collect information from the presynaptic neuron, okay. So on the basis of location, as you to see in this figure, there are three types is possible, okay. First is exodendritic, second is exosomatic, third is exogonic, okay. So exodendritic means the synapse that present between the axon to dendrites. Okay, as you have to know the structure of neuron. Okay, so this is the structure of neuron in which if there is synapse present between the axon and the dendrites. Okay, so it is called exodendritic chemical synapse, and if it present between the axon to the cell body as in the second figure, okay. So this is called exosomatic chemical synapse, okay. As you can see that here the axon link with the cell body, okay, and here is the small gap between the end of axon and the cell body, which is called exosomatic chemical synapse, and exonic term used when the chemical synapse present between axon to axon. Okay, so as in the third figure, you can see that the first neuron, uh, the axon of first presynaptic neuron link with the second presynaptic neuron. Okay, you have to see that the axon of both neuron are linked together. Okay, if this kind of condition is found, then the term use exogenic chemical synapse. Okay, so so as we have to discuss that the neurotransmitters play a very important role in the nerve impulse conduction through the chemical synapse. Okay, so first of all, we need to know what is the neurotransmitter. Okay, so neurotransmitter is a chemical substance, okay, that released while the uh, action potential arrive at the presynaptic neuron. Okay, then at the same time, the neurotransmitter release through the vesicles. Okay, so both excitatory and inhibitory neurotransmitter are present in the central nervous system and peripheral nervous system. And the same neurotransmitter may be excitatory in some location and inhibitory in the other location. Okay, so it also has been observed that certain neurotransmitter at one place act as a excitatory neurotransmitter, and at another place the same neurotransmitter act as a inhibitory neurotransmitter. Okay, so when it acts as a excitatory neurotransmitter, the nerve impulse conduction is take place. If it act as a inhibitory neurotransmitter, the nerve impulse conduction did not carry it. So important neurotransmitter include like acetylcholine, glutamate, aspartate, gamma aminobutyric acid, glycine, norepinephrine, epinephrine, dopamine. Okay, so all these are the neurotransmitter. So, if we talk about the general properties of neurotransmitter, so neurotransmitter effect can be modified either by the synthesis, can be stimulated or inhibited. Okay, so this is by the point of a mechanism. Okay, so on the basis of a mechanism, the neurotransmitter effect can be modified either by the synthesis. Okay, can be stimulated or inhibited. Okay, so when the synthesis is stimulated, the nerve impulse conduction take place. When the synthesis is inhibited, the nerve impulse conduction cannot take place. Okay, and the second mechanism is release can be blocked or enhanced. Okay, if the neurotransmitter release is blocked, the nerve impulse conduction cannot take place. If the release of neurotransmitter is enhanced, okay, so the nerve impulse conduction is take place. Okay, and removal can be stimulated or blocked. Okay, so if the neurotransmitter removed at the site of synapse, the nerve impulse conduction cannot take place. Okay, if it would be stimulated. Okay, so the nerve impulse cannot be take place. Okay, so it removal can be either can be stimulated, okay, or blocked. Okay, so both these conditions can be found while we study the nerve impulse conduction through the chemical synapse. And receptor site can be blocked or activated. Okay, so it is clear that when the neurotransmitter binds with its receptor, the nerve impulse conduction is take place. Okay, and when the neurotransmitter cannot able to bind with the receptor, the nerve impulse conduction cannot be take place. Okay, so if it is clear that when the receptor are free to bind with the neurotransmitter, the nerve impulse conduction take place. Okay, if these receptor site blocked, the neurotransmitter cannot able to bind and nerve impulse conduction cannot be take place. Okay, so that's why there are two possibilities: receptor site can be blocked or activated. Okay, so how the neurotransmitter can be act? It either act as a agonist or antagonist. Okay, so agonist terms used when the anything that enhance a transmitter's effect. Okay, so here sometimes it has been observed that certain substance can enhance the effect or neurotransmitter effect. Okay, so at that time the term used agonist. Okay, and while the other time when certain substance inhibit the action of neurotransmitter. Okay, so this at that time the term is antagonist. Okay, so there are two possibilities while we study the action or applications or use of neurotransmitter while we study the nerve impulse conduction through the chemical synapse. Okay, so as we have to discuss that small molecule are act as a neurotransmitters. Okay, and these are the acetylcholine. Okay, so if we talk about the acetylcholine, so these are neurotransmitter released by the peripheral nervous system. Okay, and the neuron that present in the peripheral nervous system and some of the neuron that present in the central nervous system also release acetylcholine. Okay, and it acts as a excitatory on neuromuscular junction. Okay, so anywhere where the acetylcholine release at the site of neuromuscular junction, it acts as a excitatory neurotransmitter. That means the nerve impulse conduction is take place. Okay, while if it is released at other site other than neuromuscular junction, it acts as a inhibitory neurotransmitter. That means the nerve impulse conduction cannot be longer carried out. Okay, and when the process is completed or the nerve impulse conduction is completed. Okay, so the same acetylcholine molecule can be uh, degraded by acetylcholine esterase. Okay, and the second types of neurotransmitter which has amino acid nature. Okay, like glutamate. Okay, and this glutamate is a amino acid. Okay, that released by nearly all excited neurons that present in the brain. Okay, and these glutamate inactivated by glutamate specific transporter. Okay, and the second is gamma aminobutyric acid, which is a inhibitory neurotransmitter for one third of all brain synapse. Okay, and like valium. Okay, so valium is a GABA aminobutyric acid which act as a agonist. Okay, So, so glutamate released by nearly all excitatory neurons in the brain, and this glutamate inactivated by glutamate specific transmitter. Okay, so this means when the action is over, this glutamate can be removed by the use of glutamate specific transporter. Okay, and gamma aminobutyric acid is inhibitory neurotransmitter for one third of all brain synapse. Okay, and valium is a gamma amino acid butyric acid, gamma aminobutyric acid, which is act as a agonist. Okay, and enhancing its inhibitory effect. Okay, so gamma aminobutyric acid act as an inhibitory neurotransmitter. Okay, so it means uh, this is particular block the nerve impulse conduction. Okay, and acetylcholine, as we have to discuss that it is released by the peripheral nervous system and some central nervous system neuron, and acetylcholine is an excitatory neurotransmitter at some synapse such as neuromuscular junction, where it binding to inotropic receptor and open cation channel. Also an inhibitory neurotransmitter at other synapse where it binds to a metabotropic receptor that open potassium channel. Okay. For example, acetylcholine slow heart rate at inhibitory synapse that made by parasympathetic neuron of the vagus nerve. Okay, so as, you know that the heart rate control by the vagus nerve, or the nerve impulse conduct to the vagus nerve. Okay, so here acetylcholine slow the heart rate. Okay, and here it act as a inhibitory neurotransmitter. Okay, and it activate by the enzyme acetylcholine esterase. Okay, and other neurotransmitter if we talk about, so these are amino acid, aspartate, glutamate, gamma aminobutyric acid. Okay, and some of the other biogenic amines also act as a neurotransmitter like noradrenaline, adenyline, dopamine, catecholamine, serotonin. Okay, so these are the some biogenic amines that act as a neurotransmitter. Okay, and ATP and other purines also act as a neurotransmitter, and nitric oxide also act as a neurotransmitter, and some of the neuropeptides also act as a neurotransmitter. Okay, so these are the other neurotransmitter. And excitatory transmission, that is the neurotransmitter receptor reaction on the postsynaptic membrane, depolarize the membrane and initiate action potential. Okay, so this is excitation or stimulation. Okay, and acetylcholine is typically an excitatory neurotransmitter at neuromuscular junction, and other than neuromuscular junction it act as a inhibitory neurotransmitter. Okay, and for inhibitory transmission, reaction between the neurotransmitter and the receptor opens a potassium channel in membrane, uh, so that potassium diffuse out of the cell, but no effect on the sodium channel. Okay, so this is the mechanism of action of inhibitory transmitter, where reaction between neurotransmitter and the receptor opens a potassium channel in the membrane. Okay, so that the potassium diffuse out of the cell membrane, but no effect on the sodium channel. Okay, so this action makes the inside of the membrane even more negative than its resting condition. This is called hyperpolarization, and it's hyperpolarize the membrane and make it more difficult to generate an action potential, and this is inhibitory action. Okay. Now, how the signal transmission take place at the synapse site. So, although the plasma membrane of presynaptic and postsynaptic neuron in a chemical synapse are close, they do not touch. And the synaptic cleft is a space of 20 to 50 nanometer that is filled with interstitial fluid that is separated to neurons. And the presynaptic neuron convert an electrical signal, that is nerve impulse, into the chemical signal by the help of release of neurotransmitter. Okay, and now the post synaptic neurons receive the chemical signal, and it generate an electrical signal again. Okay, so what is the process? First, the electrical signal convert into the chemical signal, then the chemical signal again convert to the electrical signal. Okay, so all this process take place at the presynaptic neuron, synapse, and post synaptic neuron respectively. And chemical synapse is a action potential reaches end value, and the voltage gated calcium ion channel open. Okay, and calcium ion flow inward, triggering the release of neurotransmitter. This is a mechanism of how the electrical signal that received by the presynaptic neuron convert into the chemical signal. Okay, so when the calcium ion flow inward, this calcium ion trigger a release of neurotransmitter, and the neurotransmitter crosses the synaptic cleft and binding to ligand gated receptors. And the more neurotransmitter release, greater the changes in potential of the post-synaptic cells. Okay, and the synaptic delay is about zero point 0.5 millisecond. Okay, so so it is a time which takes to convert electrical signal into the chemical signal. Okay, and it is a one-way information transfer. And a typical chemical synapse will transmit a signal as follows. So first of all, a nerve impulse arrives at synaptic end valve of a presynaptic axon, and now the depolarizing phase has to be started, and the nerve impulse open voltage gated calcium ion channel, which are present in the membrane of a synaptic end valve, and this increase calcium ion concentration inside the presynaptic neuron. Okay, which is triggered exocytosis of some of the synaptic vesicles. As vesicles membrane merge with the plasma membrane, the neurotransmitter molecule released into the synaptic cleft. Okay, now the neurotransmitter molecule diffuse across the synaptic cleft and bind to the neurotransmitter receptor in the postsynaptic neuron plasma membrane. Now, the binding of neurotransmitter molecule to their receptor, which is called or which is activate ligand-gated ion channel. Okay, now the ligand-gated ion channel open, and it allow a particular ion to flow across the membrane. Okay, now, as ion flow in through the open channel, the voltage across the membrane changes. Okay, or the membrane potential become changes. Okay, and this changes is in membrane voltage is a post-synaptic potential. Okay, and when depolarizing post-synaptic potential, this is at a threshold level. It triggers one more nerve impulse. Okay, so now the chemical signal convert into the electrical signal. Now if we talk about the neuromuscular junction, what happened at the neuromuscular junction? So the synapse between somatic motor neuron and skeletal muscle fiber is called neuromuscular junction. Okay, and the structure of the presynaptic neuron same with the other neuron. Okay, and here the neurotransmitter released in the synaptic cleft particularly is acetylcholine, and this region of the sarcolemma opposite to the synaptic end valve are called motor end plate. Okay, and within the motor end plate there is many acetylcholine receptor is present. And the neuromuscular junction, which is a motor neuron, have a threshold axon that extend from the brain or spinal cord to a group of muscle fiber. Okay, so at the neuromuscular junction, the neurons uh, uh, make or extend up to the muscle fiber, and the action potential arise at the interface of the motor neuron and the muscle fiber. Okay, and when communication occur between a somatic motor neuron and a muscle fiber. Okay, so there is a small gap which is called synapse. Okay, and the gap particularly that separate the two neuron or the neuron and muscle is called synaptic cleft, and the chemical which is released at, while the nerve impulse conduction has to take place through the chemical synapse is called neurotransmitter, and the synapse basically is a like structure that's suspended within the synaptic end valve containing molecules of the neurotransmitter that is acetylcholine. Okay, and motor end valve, the region of muscle cell membrane opposite to the synaptic end valve that contain acetylcholine receptor, which is called motor end plate. So, neuromuscular junction, a nerve impulse elicits a muscle action potential in the following ways. First of all, release of acetylcholine. So, arrival of impulse at the site of synapse, okay, causes many synaptic vesicles undergo exocytosis, and the acetylcholine liberate into synaptic cleft and diffuse between motor neuron and motor end plate. Now the activation of acetylcholine receptor, binding of two molecules of acetylcholine to the receptors on the site of neuromuscular junction, open an ion channel in the acetylcholine receptor. Once the channel is open, small cations, most importantly sodium ion, can flow in across the membrane. Now the production of muscles action potential. So the inflow of sodium ion makes the inside of the muscle fiber more positively charged, and this changes in the membrane potential trigger a muscle action potential, and the muscle action potential that propagate along the sarcolemma into the tubule system, and this causes the sarcoplasmic reticulum to release its stored calcium ion into the sarcoplasm, and now the muscle fiber suddenly contract, and the after the nerve impulse conduction has to be complete, the termination of acetylcholine activity has to be take place. Now the acetylcholine are rapidly broken down by the enzyme acetylcholinesterase, and acetylcholine now break down into the acetyl and choline, where this product can't activate the acetylcholine receptor. Okay, because the inner impulse conduction now is over. So as you can see in this figure, this is a synaptic transmission, that communication between the neurons. Okay, as you can see in this figure, there is muscle sensory receptor is there. Okay, and the information transfer through the sensory and afferent axon. Okay, and this goes into the spinal cord, as you to see in this figure. Okay, there is presynaptic neuron and the postsynaptic neuron is there, and the small gap between the presynaptic and postsynaptic neuron is called synapse. Okay, so this is the path of nerve impulse conduction. Okay, so the you can see that this figure clearly indicate that how this procedure so has to be take place. So this is the Non-impulse consumption is a one-way process, as you can see here. Okay. Now, there are two principal kind of synapses there, as we have to discuss in this lecture: electrical synapse and chemical synapse. Okay, so if we talk about the electrical synapse, so here there is a gap junction is present. Okay, you have to see in the figure A, this is electrical synapse. Okay, so in at the presynaptic neuron, there is a microtubule is present, cytoplasm containing bulb is there, and mitochondria is there. Okay, for energy generation, and here the junction is present at the site of pre and post synaptic neuron. Okay, so this is called gap junction, and you have to see that through the gap junction the electrical signal can be transmitted, and there is no need to convert electrical signal into the chemical signal, so that the electrical signal directly transmit through the electrical synapse. Okay, so the specific function of this electrical synapse is the presence of gap junction. Okay, now the second diagram shows that the presynaptic neuron and postsynaptic neuron, there is a small gap which is called synapse. Okay, and here is synaptic vesicles is there. Okay, so when the nerve impulse arrive at the synaptic bulb, okay, the calcium channel are open due to the change in the membrane potential. Now the calcium due to the calcium inward flow, this synaptic vesicle can be fused with the membrane. Okay, and the end bulb of the presynaptic neuron, and now the neurotransmitter that present in the synaptic vesicles released into the synapse. Okay, or synaptic cleft. Okay, so you have to see that clearly, see that there is a synaptic vesicle fusing, and the exocytosis is there, and the neurotransmitter is which bind with the receptor that present in the the end valve of postsynaptic neuron. Okay, now the postsynaptic neurotransmitter receptor bind with the neurotransmitter release the site of synaptic cleft. Okay, now the ion flow through the postsynaptic channels. Okay, so when these neurotransmitter bind with the uh, receptors. Okay, it open the ligand gated ion channel. Okay, which due to which this chemical signal now convert into the electrical signal. Okay, so here chemical synapse, the electric signal first convert into the chemical signal. Now the chemical signal convert into the electrical signal. Okay, so there are two kind of synapses there: electrical and chemical. So you have to see that the detailed diagram of the uh, gap junction. Okay, so the gap junction are formed where hexameric pores called connections connect with one between cells. Okay, so here you can see that these are the post synaptic cell membrane. Upper is the pre-synaptic cell membrane. These are the connections. Okay, the small channel-like structure and pore connecting cytoplasm of two neurons. Okay, so this is the special types of junction. This is called gap junction. You have to see that the electrical synapses are built for speed. Okay, and this figure shows that how the membrane potential change at presynaptic neuron and the postsynaptic neuron over the time of millisecond. So the electrical synapse are not presently considered to be the primary means of communication between neurons in the mammals nervous system, but they may prove that the more important than presently recognized. And spiking activity among cluster of neurons. The most thoroughly studied electrical synapse occur between excitatory projection neuron of the inferior olivary nucleus and between inhibitory interneurons of the neocortex, hippocampus, and thalamus. And all these synapses require the game junction where the protein is present, which is called connexins. Okay, connexins 36. For robust electrical coupling, the connexins 36 appear to interconnected neurons exclusively, and it is expressed widely along the mammal's neuroaxis, and implying that there are undiscovered electrical synapses throughout the central nervous system. and some central nervous system maybe has electrical synapse. So, if we talk about the rectification and unidirectional of electrical synapse, so not just a simple bidirectional bridge between the cells, and conductance through gap junction may be sensitive to the junctional potential, that is the voltage drop between the two coupled cell, or sensitive to the membrane potential of either of the coupled cell. And here the glial cells, okay, so glial cells can also be connected by the gap junction, and these glial cells allow synchronous oscillation of intracellular calcium. So this figure clearly shown this what happened at the site of chemical synapse. Okay, so here you have to see that the when the nerve impulse arrive at the site of pre-synaptic neuron. Okay, here you have to see that at this point. Okay, so an action potential invade the presynaptic terminals here. Okay, this one. Now the transmitter is synthesized and then stored in the vesicle. Okay, before the uh, time of action potential that invade the presynaptic terminals. Now, when the action potential invade the presynaptic terminal, the depolarizing of the presynaptic terminal cause opening of voltage gated calcium channel. Now the voltage gated calcium channel open due to the change in membrane potential due to the action potential arrive at this site. Okay, so due to the change in membrane potential, the calcium channel are open. Okay, so this is voltage gated calcium channel. So now this voltage gated calcium channel are open and inward flow of calcium ion has to be started. Okay, and due to the now the calcium cause vesicles to fuse with the presynaptic membrane. Now you can see that here the vesicles, synaptic vesicles, which has transmitter neurotransmitter molecules fused with the membrane here. Okay, so when this presynaptic this vesicles fuse with the membrane, which release neurotransmitter at the synaptic cleft. Now the transmitter is released into the synaptic cleft via exocytosis. Okay. Now, this neurotransmitter bind with the receptor molecule in post-synaptic membrane. Okay, and it leads to the opening or closing of a post-synaptic channel. Okay, now the postsynaptic current causes excitatory or inhibitory postsynaptic potential that changes the excitability of the postsynaptic cells. Okay, so now you have to see that now here you have to see that the electrical signal which is arrived at the site of synaptic bulb convert into the chemical signal. Now the chemical signal convert into the electrical signal. Okay, so this process occur at the chemical synapse, and there are certain criteria that define a neurotransmitter. Okay, so it must be present at presynaptic terminals. Okay. Now there are certain criteria that define a neurotransmitter. So must be present in presynaptic terminals and must be released by depolarization and calcium ion dependent. Okay, it must be calcium ion dependent and specific receptor must be present. Okay, so these are the certain criteria of neurotransmitter. Okay. Now Now the neurotransmitter may be either a small molecule or peptides. Okay, here that you have to see that the mechanism and site of synthesis are different. Okay, so the small molecules transmitter are synthesized at terminals and packaged into a small clear core vesicles, which is called synaptic vesicles. Okay, and peptides or neuropeptides are synthesized in the endoplasmic reticulum and transported to the synapse, and sometimes they are processed along the way. uh, okay, neuropeptides are packaged in a large dense core vesicles. Okay, here you can see that these are the small molecule neurotransmitter and these are the peptide neurotransmitter. Okay, so there are certain difference between the synthesis of these neurotransmitter. Okay. Now the phase which is occur during the nerve impulse conduction, which is called synaptic release. Okay, so the synaptic basically release cycle. The first one is tools and pools, and second one is molecular biology and biochemistry of vesicles release, which involve docking, priming, and fusion. And third one is recovery and recycling of synaptic vesicles. So in this way, you to see that all these processes. Okay. So first of all, you have to see that this is endosome. Okay, and it will form a vesicle through the budding. Okay, now the docking, priming, and fusion has to be take place. Okay, it you have to see that it would occur within 1 millisecond. Okay, and 10 to token 20 second it to take for endocytosis. Okay. And how do we study vesicle dynamics? Okay, so the first is morphological techniques. Okay, it include electron microscopy to obtain aesthetic picture of vesicle distribution. Okay, and the total internal reflection fluorescence microscopy to visualize movement of vesicle close to the membrane. And the physiological studies include chromaffin cells, which include endocrine cells derived from adrenal medulla with large dense core vesicle, and can measure membrane fusion. Okay, or the direct release of catecholamine and transmitters using carbon fiber electrode, and third one is neurons that measure release of neurotransmitter from pre-synaptic cells by quantifying the response of a post-synaptic cells. Now the genetics include delayed or overexpressed protein in mice, worms, flies, and analyze phenotype using the above techniques. Okay, so this is the way how we can study the vesicles or synaptic vesicles dynamics. And the steps while the synaptic vesicle fuse at the membrane, it include docking. Okay, docking means docked vesicles lies close to plasma membrane within 30 nanometer uh, area around. And priming, where primed vesicles can be induced to fuse with the plasma membrane by sustained depolarization. And high potassium ion, elevated calcium ion, hypertonic sucrose treatment. And the fusion include the vesicles fused with the plasma membrane to release neurotransmitter. And physiologically, this occur near the calcium channel, but can be induced experimentally over larger area. Okay, so the active zone is the site of physiological release and can sometimes be recognized as an electron dense structure. So in this figure, you have to see that all these processes. Okay, so vesicle release require many protein on vesicle and plasma membrane. Okay, so it is very complex process. So docking include UNC18, which is necessary for vesicle docking. Okay, and UNC18 mutant C elegans have neurotransmitter release defect. Okay, so this shows that UNC18 is necessary for vesicle docking, and UNC18 mutant C elegans have reduction of docked vesicles. This shows that UNC18 has important role in the docking of synaptic vesicles. Okay, you have to see that this is the process. Okay, budding. Okay, and docking. Okay, and now fusing. Okay, priming, now fusing. Okay, so the priming is the process where the vesicles in the reserve pool undergo priming to enter the readily releasable pool. Okay, which is ready for release their content and fuse with the membrane at the site of synaptic and bulb. And at molecular level, priming corresponds to the assemble of snare complex. Okay, and what is the function of priming? So MUNC13 is required for priming. Okay, you have to see that in this figure. What happened at the time of priming? Priming means it is ready to fuse with the membrane. Okay. Now the synaptogemin function is a calcium sensor and promoting vesicle fusion, and synaptic vesicle recycle post fusion. Okay. Now, endocytosis retrieves synaptic vesicles membrane and protein from the plasma membrane uh, after fusion. Okay, so as you can see that in this figure, the first of all adapter protein connect kilethrine to vesicle membrane. Now, kilethrine triskadia assemble into coat curving membrane. Now, the dynamics ring form and pinches of membrane. Now the coated vesicle translocated by actin filament. Now the ASC17 and oxygen protein uncoat vesicles. Okay, so this is the process of endocytosis. Now you have to attempt quiz. The first question: anything that enhances effect of a neurotransmitter is called. Okay, so this is called agonist. Okay, so anything that enhances the effect of neurotransmitter, any substance. Okay, so the option A is correct. And anything that block the action of neurotransmitter. Okay, so while any substance that have potential to block neurotransmitter effect is called antagonist. Okay, and at neuromuscular junction, neurotransmitter acetylcholine act as excitatory neurotransmitter only. Okay, so it is at neuromuscular junction. It is clear that we have to discuss in this lecture. It acts as a excitatory neurotransmitter. So the option A is correct. And the enzyme acetylcholinesterase act as inactivate acetylcholine, activate acetylcholine. Okay, so it is clear that when the action is over, acetylcholinesterase broken down acetylcholine into the uh, into the acetylene and choline. And the potential used for short distance communication, that means this is a graded potential. And the potential used for long distance communication, it is action potential. Okay. Thank you everyone.